drug-resistant infections, or AMR as scientists call it. Well, it's not some far-off problem anymore, is it?
No, not at all. It's a full-blown crisis. We're talking over 1 million deaths globally each year right now.
And today, we're doing a deep dive into really one of our last lines of defense against some of the toughest bugs, the polymyxins.
That's right. These are the, quote, last resort antibiotics. We use them when pretty much everything else has failed.
Specifically against gram-negative bacteria.
Exactly. And when you hear gram-negative, you should think armor. Serious armor. They have this incredibly tough outer membrane, like a biological shield.
Which makes them just impermeable to lots of common drugs.
Precisely. It keeps many antibiotics out.
So, okay, we know polymyxins work somehow by, getting through that armor. But for decades, it's been a bit of a black box, hasn't it?
It really has. I mean, these drugs were discovered, 80 years ago? We knew they hit the surface, but the actual how, well, that was fuzzy.
And that's the mission for this deep dive then. We're going to unpack some really groundbreaking research. Came out of a big collaboration, UCL, Imperial College London.
Some major UK institutions, yeah. They used some seriously advanced imaging tech to finally see exactly how these drugs launch their attack.
And crucially, they also figured out why they sometimes fail, which explains those really dangerous recurrent infections people can get.
Yes, and understanding that failure mechanism, that's become an urgent priority. This isn't just academic curiosity, it's vital for global health. Finding this out after 80 years is, well, quite the breakthrough.
Okay, let's kick off with the visuals then, because you mentioned imaging, and apparently it's pretty stunning stuff. They needed something much more powerful than a regular microscope, right?
Oh, absolutely. You're looking at things on a nanoscale. They used a technique called atomic force microscopy, AFM, over at the London Center for Nanotechnology.
AFM. How does that work? It's not using light.
No, not light. Imagine an incredibly tiny needle, just a few nanometers wide. It literally scans across the surface of the bacterial cell. It sort of feels the shape, the bumps and dips.
Like reading Braille on a microscopic level.
That's a great analogy, actually.
Yeah.
Yeah, it builds up this topographical map. The images have unbelievable clarity. You can see features measured in nanometers. It's quite amazing.
And they took pictures over time, like a time lapse.
Exactly. They captured these composite images showing the bacterial surface at 15 minutes, 30, 60, 90 minutes after adding the polymyxin.
And what they saw wasn't the armor just dissolving away?
Not at all. It was something far more dramatic and totally unexpected. It looked like, well, like bacterial self-sabotage.
Self-sabotage.
Yeah.
Okay, so let's get slightly technical for a second. Polymyxins are positively charged molecules, right?
Huh.
And the key component of that gram-negative armor, the stuff giving it strength, is called lipopolysaccharide, or LPS, and it's negatively charged.
So opposites attract. The drug sticks to the LPS. Precisely. It binds really strongly. But what the AFM images showed is that this binding doesn't just poke a hole, it triggers a kind of panic response in the bacterium.
A panic response.
Yeah, the cell senses this damage, this polymyxin, stuck to its LPS, and it kicks its repair systems into overdrive. Frantic, high-speed repair.
So it tries to fix the damage by building its wall faster.
Much, much faster. Way too fast, actually. The antibiotic forces the cell to just churn out and shed its outer layer components at such a ridiculous rate that the whole structure just destabilizes.
Well,
You literally see these bulges and bumps appearing on the surface in the images, and then boom, catastrophic holes rip open in the armor.
that's just wild.
Yeah.
So the antibiotic isn't the direct killer in that sense. It tricks the bacteria into destroying itself with its own building machinery.
That's the core insight we've been missing for 80 years. It forces this overproduction. Carolina Borelli, one of the co-authors, used a really good analogy. She said it's like forcing the cell to make new bricks for its wall so incredibly fast that the wall itself just collapses under the chaotic construction.
So the integrity is lost because of chaos, not slow decay.
Exactly, self-generated chaos. And once those holes open up, the polymyxin can get inside and finish the job, killing the cell from within.
Okay, so that explains the power, this violent self-destruction.
Yeah.
But you mentioned there was a twist, something that explained why these powerful drugs sometimes fail in hospitals.
Yes, this is where it gets really, really interesting and frankly, quite concerning for treatment.
What is it?
It turns out that this whole dramatic process, the bulging, the super fast production, the shedding, the eventual death, it only works if the bacterium is actively growing and replicating.
Really when it's active.
Only then. If the bacterium is dormant, basically sleeping,
Yep.
none of that happens.
So the drug needs the cell to be, like, in construction mode
Yeah.
for this self-sabotage trick to work.
You've got it. Dormancy is a survival state. Bacteria use it when things get tough, typically when there's no food around, a lack of nutrients.
Like hibernation for bears, but for bacteria.
A very good comparison. And when they go into this dormant, sleeping state, that whole armor production and repair factory, it gets switched off, completely shut down.
That feels like a massive blind spot we've had. I read that Dr. Andrew Edwards at Imperial mentioned the assumption was always that these armor busters would kill bacteria no matter what state they were in. Active, dormant, didn't matter.
That was the decades-long assumption, yes. Because, it attacks the structure itself, so why should the cell's activity level matter?
Right.
But this research flips that. The drug isn't just passively dissolving the wall, it's actively hijacking the cell's own machinery. If that machinery is off because the cell is dormant,
Then the hijacking fails.
precisely. The drug still sticks to the outside, to the LPS, the AFM showed that, but it just sits there. It doesn't trigger that fatal overproduction cascade. It might cause some surface damage maybe, but it doesn't kill the cell.
And connecting that back to the listener, thinking about patients. This is huge for those recurring infections, isn't it?
It absolutely is. Think about it. You get a serious infection, you take the polymyxin, it wipes out all the active growing bacteria that are causing the immediate symptoms. You feel better.
Sure.
But hiding in your system might be a population of dormant cells. The sleepers, they weren't actively building their armor, so the antibiotic course just washes right over them. They survive. Then, maybe days or weeks later, conditions improve. Maybe your nutritional state gets better or the environment changes somehow. The bacteria sense it's safe. They wake up.
And the infection comes roaring back.
Exactly. A dangerous recurrent infection, potentially caused by bacteria that we're already exposed to and survived our last resort drug.
That also makes me wonder, if dormancy is triggered by a lack of food, does that mean really sick patients, maybe malnourished ones, could have more dormant bacteria, making their infections even harder to treat?
That's precisely the implication. It's a critical link. The patient's own metabolic state, their environment, could directly influence how well the antibiotic works by affecting the bacteria's state. It's not just about the drug and the bug anymore. It's the drug, the bug, and the host environment.
Wow. Okay. So how did the researchers actually prove this link between dormancy and the drug failing? They must have run a specific test.
They did, a really neat one. They took E. coli, a common gram-negative bacterium, and set up two groups. One group was actively growing, well-fed. The other was deliberately starved to push it into a dormant state.
And then they added polymyxin B to both.
Yep. And the results were crystal clear, exactly as the imaging predicted.
What happened?
The active growing cells, wiped out, efficiently killed. The self-destruction mechanism worked perfectly.
And the dormant ones.
Completely untouched. The antibiotic didn't kill them at all. Now, importantly, they checked, and the drug was binding to the surface of these dormant cells. It was stuck there.
But it just couldn't pull the trigger for self-destruction because the cell's machinery was off.
Exactly right. It was physically present, but functionally useless against a dormant cell.
Okay, so if the drug only works when the cell is active and building, the logical next step would be to try and wake the dormant cells up while the drug is still there, right?
That's the crucial experiment. How do you wake up a dormant bacterium?
Give it food.
Give it food. They introduced simple sugar glucose into the culture dish containing the dormant E. coli that already had polymyxin bound to their surface.
Did the sugar suddenly make the antibiotic work?
It did, but with a fascinating detail. The killing didn't start instantly. There was a delay, a very precise delay of about 15 minutes after adding the sugar. Only then did the bacteria start dying.
15 minutes. Why that specific time?
That's the beautiful part. That 15-minute window is almost exactly the time it takes for a dormant E. coli to sense the sugar, fire up its metabolism again, start actively producing its outer membrane component.
And basically make itself vulnerable to the polymyxin that was already sitting there waiting.
You nailed it. That delay is the smoking gun. It proves unequivocally that the bacterium's metabolic state, active versus dormant, is the absolute deciding factor in whether polymyxin works or not.
That's incredibly clear proof. So knowing this mechanical weakness, this dependency on the bacteria being active, what does this mean for actually treating patients better? Professor Bart Hugenboom mentioned needing a kind of twin approach.
Yes, this is where we pivot from understanding the problem to potentially solving it. The old way was just hit the bacteria hard. The new way has to be smarter. It points towards combination therapies.
But hang on, isn't there a risk? If you're deliberately waking up dormant, dangerous bacteria just so you can kill them, aren't you potentially making things worse for a short time, increasing the active bacterial load?
That's the absolute crux of the challenge, and why any strategy like this needs to be incredibly carefully designed and timed. You're right, just waking them up indiscriminately could be dangerous.
So how would this twin approach work safely?
The idea is not just to wake them up, but to wake them up while the polymyxin is already there, bound and ready, or at a high enough concentration. The combination would involve the polymyxin plus a second agent.
Okay, and that second agent could do one of two things, you said.
Right. The first possibility is maybe the most counterintuitive one.
Go on.
You could, theoretically, use a second drug that actually promotes the bacteria's own armor production.
Wait, help it build its wall. Why would you do that?
Because if you can artificially kickstart that building process, even in a dormant cell, or push an active cell to build even faster, you trigger the self-destruction cascade. You force the chaos that polymyxin relies on.
Huh. Okay, that is counterintuitive. And the second function for the partner drug?
That's the more obvious one. Find a safe way to be the alarm clock. Find an agent, maybe something related to sugar, maybe something else entirely that targets bacterial metabolism to reliably wake up the sleeping bacteria in a patient.
So you ensure all the bacteria, active and dormant, are awake and building their walls before or while the polymyxin is doing its work.
Exactly. You prepare the battlefield, make sure all targets are vulnerable, then hit them. It really pushes antibiotic development towards thinking about metabolism.
And this must also change how we even look for new antibiotics in the first place, surely. We usually test drugs on happy, well-fed, actively growing bacteria in a lab dish.
That's been the standard model for decades, and this research shows just how inadequate that can be. It completely misses the dormancy loophole.
So future drug screening needs to test against both active and dormant bugs.
It absolutely has to. If you want to know if a drug will truly eradicate an infection long-term, not just knock it back temporarily, you need to know if it can handle dormant cells. This collaboration between UCL, Imperial, Nottingham, it's really provided a fundamental insight.
Okay, so let's recap this deep dive then. Polymyxins are last resort antibiotics. They work not by simply dissolving bacterial armor.
No, they work by forcing the bacteria into this state of frantic, chaotic overproduction of that armor, leading to self-destruction, like making it build its own prison walls so fast they just collapse.
But the huge catch, the Achilles heel, is dormancy. If the bacteria is asleep and not building.
The drug is basically useless. It needs the cell to be active, which explains why infections can seem to clear up, only to return later.
This research has basically given us the instruction manual for polymyxins after 80 years, and crucially, shown us the flaw we need to fix.
Precisely. Understanding the why of failure is the first step to overcoming it.
Which leads to a final thought for you, our listeners, building on what we've discussed. If waking up dormant bacteria is key, and we saw it works with something as simple as sugar in the lab, what else could work in a real patient?
Yeah, think beyond just simple sugars. What about other non-antibiotic approaches? Could we manipulate a patient's metabolism subtly? Or maybe even use specific components of diet? Or perhaps interventions targeting the gut microbiome to gently nudge those dormant bacteria awake?
So they become vulnerable to the polymyxins we already have, combining metabolic nudges with our last resort drugs.
That's the frontier, isn't it? Finding safe, effective ways to ensure all the harmful bacteria are metabolically online when the antibiotic arrives, that's where the synergy might lie for truly beating these resistant infections. Something to definitely think about.